Cretaceous dinosaurs and birds was conducted by Fusion Zhang, Stuart Kearns, Patrick Orr and fellow colleagues. The aim of this study was to determine the evolutionary relationship between non-avian theropod dinosaurs and modern-day birds through the discovery of melanosomes found in fossilised feathers. Then, from the results, to provide a further understanding of what colour dinosaurs may have been and an insight into the function of feathers. The methods used to perform tests on each of the fossils were carried out by using a Hitachi variable pressure scanning electron microscope, equipped with an energy dispersive spectrometer. The samples were individually mounted on pin stubs using double-sided carbon tape. Figure 1, the isolated pennaceous feather, was a basic test examined under SEM. Figure 2, the Confuciusornis sample, was from the periphery of the specimen ranging in size from 3 to 300 mm squared. Figure 3, Sinosauropteryx, an undescribed dinosaur specimen, a flake sample was removed from the dorsal side close to the base of the tail. Figure 4, Sinomythosaurus samples were taken from the surface of the holotype. This graph simplifies the results, displaying how each fossil sample contains melanosomes and further verifying the two types, eumelanosomes and phamelanosomes, due to distinct shapes and sizes. Similarly, this table sums up the samples and their results, as well as additional images. Proven with 3 pieces of evidence, this strategic study confirms that the microstructures in the Joel fossils are in fact melanosomes, not replacement bacteria. Firstly, the melanosomes found embedded inside the fossils are similar to those in modern birds, as extant birds have evident feather barbules arranged in complex arrays that are also present in the preserved Joel fossils. Secondly, it was discovered that dark bands of banded feathers only occur in eumelanosomes, not in light bands. A reference in the study provides the third piece of evidence to support the existence of fossilised melanosomes. It showed packing and layering of melanosomes in fossil feathers which are identical to ultrastructures in modern feathers and in Joel feathers, as well as being incompatible with bacteria origins. Due to the occurrence of melanosomes in fossil birds and non-avian dinosaurs provides the opportunity to reconstruct certain aspects of the external coloration of these organisms. By identifying both eumelanosomes and phamelanosomes, the authors can imply that some basal birds and non-avian theropods had black and russet coloration. The identification of different melanosomes in the fossilised dinosaur feathers helps to identify and suggest how they were coloured, and further to discover the functions of the feathers in non-avian dinosaurs by reconstructing colour patterns, either primarily for thermoregulation, camouflage or communication. Melanosomes are microbes, testing an alternative hypothesis for the origin of microbodies in fossil feathers, was published by Alison Moyer, Wexina Zhang, Elizabeth Johnson and fellow authors. Due to the similarity of the morphology of melanosomes and microbodies, both hypotheses were re-evaluated only to discover a microbial origin, meaning there is a clash between studies, where both have been proven plausible.